Real-life mutants are kinda neat, but nowhere near as dramatic as TV. Yeah, mutations can come from exposure to radioactive materials and chemicals, and yeah, you can be born with it, and you can be infected with something that gives you mutations. But do any of these mutations really give superpowers? Well, no, not really. Unless there's a gene that encodes for very localized kinetic dimension portal openings in the eyes, or genes for manipulating the very laws of thermodynamics, then scientists have yet to find it. What scientists have found is that a lot of mutations can lead to genetic diseases, deformities, aging, and cancer. A lot of cancer. Usually cancer. So, mutations are often bad, but not all. Sometimes they're harmless and you have a weird base pair in a spot that doesn't encode for anything, so you're fine. Sometimes they do something and it doesn't really hurt you, so you're fine. Sometimes those mutations take a while before they become a problem, so you live normally for a time. And sometimes those mutations set you on an evolutionary path that makes you really well suited for specific circumstances in which having that quirk about you is what allows you to survive and pass your genes to the next generation. Which is nice. But what are mutations? Well, they're changes in the genome that results in different versions of DNA from the usual. Mutations can come from viral infections, outside, like the sun, and inside. Well, not always from the food you eat, even though they can be kind of ambiently radioactive, but from the gut microbiome. Luckily, your guts have a high cell turnover rate, so it's not a big deal. But also inside because of replication. DNA mistakes in replication are pretty rare. 1 in a billion. Very nice when we've got about 3 billion base pairs in a human genome and there's about 37 trillion cells in a given person. Mutations can come from mismatches, whoopsies, weirdo bases, more specifically, chemistry being problematic, backbone breaks, radiation, and pyrimidine dimers from UV light. Sun radiation. There's a couple of ways that DNA can be fixed, like photolyase. Sometimes the sunlight, UV light, makes thymidines stick together. The TT sticking together distorts the DNA. Photolyase unsticks them with light. Photo means light, lysate means to break. Light-breaking enzymes. By the way, only prokaryotes get to do this. We just get sunburnt or melanoma. Alkyltransferase. A whole molecule that's dedicated to removing this off from where it's not supposed to be. Let's say this is O6-methylguanosine. 6 is the position of the oxygen of interest, though given that guanosine has the one O, whatever. This is methyl, that one, with the CH3 sitting on the oxygen. It makes the guanosine thinks it's an adenine, so it's going to try to pair with a T instead of a C, and that's bad. So alkyltransferase transfers this alkyl off the O on the guanosine so it can go back to pairing with the proper C. And that's it. Now on to clipping it out and doing it over. NER, nucleotide excision repair. UVR-ABC endonuclease looks for the problem spot and cuts the mistake out. UVRD twists the DNA to pop it out and pol-1 and ligase fills in the gap. BER, base excision repair. Glycosylase snips the glycosidic bond, glyco, sugar, and acidic bonds referring to the nucleic acid with ribose sugars, leaves an AP site. AP meaning apyrimidic or apurine, A meaning without, a base site, and leaves a spot for pol-1 and ligase to fill it in proper. Mismatch repair. Mu-S and Mu-L bind to the problem spot to create a big loop up to 1,000 base pairs big, and uses Mu-H to snippy snip here, an exonuclease to shear the loop up to the problem spot and has pol-3 and ligase fill in the gap. Why pol-3? Because the exonuclease shears off from 5' to 3' section, and there's a lot of nucleotides to fill in again. Now on to double-strand breaks. What do you do when you don't have a clear idea of how stuff goes back together? 2 stray ends that need to get connected somehow. Well, we've got 2 ways. Homologous and non-homologous. Think of cell replication having a schedule of sorts. There's a time that DNA has to be ready before anything else can progress to the next step. DNA is the manual of everything a new cell is going to need for it to become what it is, as well as everything for all the other cells it'll split off into. Now say you ripped that manual into pieces. That's the equivalent of a double-strand break. So how would you go about fixing it when you're under crunch time? Well, option 1 is slamming them together and getting it out. This is non-homologous recombination. Q70 and Q80 take separate ends of the double-strand break, MRE11 clips the ends so they can come together, Q70 and 80 brings the pieces together and close for XRCC4 with ligase 4 to stick the ends together and fixes the break. The problem with non-homologous is that it's pretty much always mutagenic. It's like taping a torn page somewhere in another ripped page in the book. Depending on what's messed up in what cell, it might not be a big deal. Like, if you messed up the instructions to making melanin in the liver, do you think it's really gonna give a damn? You think this idiot cares that much about making UV protective pigment? While in other areas like hair, yeah, losing the instructions to making melanin kinda sucks. One aspect of aging is the accumulation of these non-homologous sort of fixes. Skin and hair are plain to see, and due to their high cell turnover rate and regular exposure to the elements, they might run into mutations sooner than other cells. I might be able to discuss aging another time if people are interested. Moving on, there's another option to fixing double-strand breaks, and that's comparing with a completed copy, known as homologous recombination. And, I think I want to save this last one for its own video, as it's a bit more complicated. So, closing thoughts. It's good to understand how DNA is repaired as the first stepping stones to potentially preventing and/or treating some forms of cancer and genetic diseases. For non-biochemists, wear sunscreen and don't do the things this man did to his balls.
But Randy used a microwave, and those are non-ionizing, so they don't damage DNA.
Huh, just a microwave?
Yeah, those just warm up the water inside of things. It'll sterilize them, making them too hot, but it isn't the kind of radiation that does anything to DNA.